Here we go. Hello, everyone. My name is Aiden, and this is my individual presentation for Genomics 5. Today, I will be discussing a large-scale exome sequencing study that analyzes nearly half a million UK Biobank participants to uncover genetic links to human traits and diseases. Study overview. The article I'm presenting is titled Exome Sequencing and Analysis of 454,787 UK Biobank participants, published in Nature in 2021 by Bachman and colleagues. The goal of the study was to identify rare genetic variants in protein coding regions and investigate their associations with diseases and traits. Background on exome sequencing. Exome sequencing targets just the exomes, or the parts of our DNA that actually code for proteins, about roughly 1 to 2% of genomes. This approach is useful because most disease-causing mutations occur in these regions, and it's also a lot more cost-effective and manageable than sequencing the entire genome. Omics context. In biology, omics refers to fields that examine sets of molecules like genomics or genes, transcriptomics for RNA, and proteomics for proteins. This study fits under genomics and more specifically exomics, since it mainly focuses only on protein coding regions. The UK Biobank dataset. The UK Biobank dataset is a major research resource with nearly 500,000 participants who have provided genetic, health, and lifestyle data. This makes it one of the largest and richest biomedical datasets available today, which is very ideal for population-level genomic studies. Study design and methods. Researchers examine exome capture methods combined with high throughput sequencing to analyze coding regions. After identifying variants, they use statistical tools to associate these genetic changes with thousands of health and lifestyle traits recorded in the UK Biobank. Key findings. The study identified 4.7 million coding variants. Notably, over 98% of these were rare, meaning they had a minor allele frequency below 1%. They found strong associations between 231 genes and a lot of different health conditions, from cardiovascular disease to cancer. Highlighted results. Here's one of the visual highlights, a Manhattan plot showing gene disease associations. For example, variants in the BRCA2 gene were linked to increased breast cancer risk, reinforcing known findings and showcasing the study's power to detect meaningful associations. Scientific impact. This is the largest exome-wide association study ever conducted. It demonstrates how rare variants contribute to common diseases, which has a big impact for research, screening, and even future drug development. Clinical relevance. Clinically, this work supports precision medicine, identifying rare variants tied to disease that can help doctors predict risk, treatments, and even identify new drug targets for more effective interventions. Strengths and limitations. The study's strengths include its massive scale and comprehensive health data. However, one limitation is its focus only on a largely European population, which which may limit its relevance. Also, interpreting rare variants remains a challenge. Personal reflection. What struck me most was the massive scale. Sequencing nearly 500,000 people is quite frankly remarkable, but it also made me think of how results could be different in more diverse populations. This opens the door to more inclusive research going forward. Conclusion: to wrap things up, this study shows how powerful exome sequencing can be for uncovering rare, disease-related variants. Large biobanks like the UK Biobank are essential for moving genomic medicine forward. This work sets the stage for future global efforts. And here's the reference for the article. Thanks so much for listening.